This is podcast 9. So far, the first 2 weeks we've talked about macromolecules. Those are lipids, proteins, nucleic acids, carbohydrates. We talked about general structure of membranes and how the fluid mosaic membrane means that membranes have different components to them: proteins, lipids, and carbohydrates. That's what makes them mosaic. And then all those parts move. That's what makes them fluid. And then we talked about membrane transport systems, how you get something across the membrane. Today, we're just going to review in general what whole cells look like and the parts, what the parts do for function. This is a review. You've seen most of this stuff before, and we'll talk a little bit about the origin of cells. So Stanley Miller did a famous experiment in the 50s where he set up a blown glass apparatus that was self-contained that was thought to simulate early Earth's atmosphere, in that it had the gas composition in the atmosphere of early Earth. It had lightning as simulated by an electrical discharge. It had heat, and it had a reflux system so that things would go up in the atmosphere, fall out of the atmosphere back into the ocean, and then be recycled. It's now thought that the conditions he used to start this experiment are not what early Earth really looked like. The chemical composition is now thought to be different. However, his results, meaning that he could make early components of life, biological molecules such as adenine, for example, and amino acids, that has been repeated over and over again in subsequent experiments. And so it seems like a plausible hypothesis that these inorganic forces, heat, electricity, recycling of elements through their different phases, could have resulted in early biological compounds in early Earth before life. It's also that some of these compounds, such as amino acids and nucleotides, have been found on meteorites. So it's possible that meteorites seeded early Earth with biological compounds. One of the hypotheses about first cells comes from RNA. This is called the RNA world hypothesis. This idea is that RNA is a precursor to DNA, and that RNA can act both as an enzyme and as a carrier of generational information. Generational information meaning that the sequence of the RNA carries information, and that sequence of RNA can be repeated from one generation to the next. So you have that characteristic, which is we think of as a DNA characteristic, is also an RNA characteristic. And then RNA also has the additional characteristic in that it can act like an enzyme. So it's possible that RNA was one of the original molecules in cells. These primitive cells would have been a liposome, a lipid bilayer that sets up spontaneously in a hydrophilic environment, in a water environment, and that the RNA becomes encapsulated inside this lipid environment, and now you've got an inside and an outside, and because the RNA can catalyze reactions, you can build up reaction byproducts inside the cell that is different from the concentration outside the cell. Some support for that is that even in systems like a ribosome that makes protein, synthesizes protein, the ribosome is made up of both RNA and protein, not just protein. And not only that, but if you remove the protein from the ribosome bit by detergent extraction, so that you end up with just RNA left on the ribosome RNA part of the ribosome can still catalyze a peptide bond. So it's the RNA that is putting proteins together, and because RNA can do double duty to carry the nucleic acid information from one generation to the next and catalyze reactions, it's thought that it's a good candidate for one of the original molecules in the original cells. This is Dr. Aaron Goldman. He's at Oberlin, and he's an expert on early life, how life got started on the planet. He is a principal investigator on this Leukopedia project. And this is the idea of this project is to find representatives of genes that are known across all taxa. So the idea here is that if we find out what genes are the oldest, in that everything on the planet has those genes, it's likely that those genes are some of the first genes in the first cells, and we can get an idea of what the first cells could do genetically, and what functions some of the first cells had. That's the Leukopedia project. Another idea that comes out of this project is that the idea of protein and protein cofactors are remnants of our evolutionary history, where perhaps originally there was catalysis of a biochemical reaction on a surface of a mineral. This is well known on some clays and some other rocks, that they can act as catalysts for biochemical reactions, make reactions more easily, more probable. They occur faster and more likely to occur. Then you have proteins coming along that carry some of that mineral surface with them in the form of, say, a porphyrin ring with a heme in it that carries a metal in the middle of that structure, and that is sort of getting the geochemical catalyst off the surface of a mineral, and now it can be in solution because it's attached to a protein. So there's this idea that proteins evolved as a way to catalyze reactions off the surface of a geochemical feature like a rock, and now in solution. And perhaps was another big step in the evolution of earliest cells. Speaking of earliest cells, when I was in undergrad, there was just bacteria and eukaryotes, so prokaryotes and eukaryotes. We now recognize that there is a third group, the archaeans, and in this very primitive tree, the ancestral cells split off, and bacteria are significantly different than archaeans and eukaryotes, and that archaeans and eukaryotes are more closely related to each other than they are to bacteria. So way back in the 80s, when I was a student, there was just bacteria versus non-bacteria. Now we recognize archaeans and bacteria as separate things. Archaeans are unicellular and thought to be closer to us evolutionarily than are bacteria. So what are characteristics of these three domains of life? Both archaeans and bacteria are small, and eukaryotes tend to be bigger in terms of the typical size of a eukaryotic cell versus a typical size of an archaean cell. Bacteria and archaeans don't have a true nucleus, don't have any membrane-bound organelles, whereas eukaryotes do. Bacteria don't have microtubules and microfilaments, but they have other proteins that serve similar functions and have similar shapes. Same for archaeans, whereas eukaryotes have true actin and tubulin proteins for forming the cytoskeleton. And we'll talk more about those later on this week in the podcast. There is no exocytosis or endocytosis in bacteria or archaeans, but there is in eukaryotes, and we'll talk more about that when we talk about endomembrane system. Bacteria can have a cell wall. Not all have a cell wall, but can have a cell wall. And so our archaeans, the composition of that cell wall is different between archaeans and bacteria. And eukaryotes can have a cell wall. Plants have a cellulose cell wall. Fungi have a cell wall made of chitin. Animals and protozoa don't have a cell wall. Bacteria divide down the middle, binary fission, and eukaryotes use either mitosis or meiosis. In bacteria, the chromosome is usually circular and is not heavily associated with proteins, but archaeans, although it is also circular, is associated with histone-like proteins. These are DNA packing proteins that we also have, and this piece of evidence is a big contribution to why we think archaeans are closer to eukaryotes than bacteria are to eukaryotes. The RNA, after it gets transcribed, is very minimally processed in bacteria, whereas in archaeans and in eukaryotes, less so in archaeans and more so in eukaryotes, the RNA is processed a little bit or a lot. So what is RNA processing? For example, cutting out the introns and putting together exons, putting together exons in different order, and then also editing the mRNA before it gets translated, single bases of the mRNA before it gets translated. Those are all characteristics of archaeans and eukaryotes, but not typically in bacteria. The initiation of transcription is different in bacteria than it is in archaeans and eukaryotes. And again, archaeans and eukaryotes share a lot of their characteristics of how they start transcription. And that's another big piece of evidence for why archaeans and eukaryotes are thought to be more closely related to each other than they are to bacteria. The RNA polymerase, we'll talk about that in a couple of weeks. What type of RNA polymerase? This is the enzyme that makes RNA from DNA. Is quite a bit different in bacteria versus eukaryotes, and in archaeans, it's someplace in between. The size and shape and protein content of the ribosomes is different in the three taxa, the three domains. Starting translation, turning a mRNA transcript into protein, is distinctly different in bacteria, but archaeans and eukaryotes it's similar. And the membrane phospholipid composition is different among the three taxa. So that's a long, boring table, but the take-home message is that there's several pieces of evidence that indicate that even though archaeans look to us when we're looking at them under a microscope like bacteria, they're much closer to a eukaryote cell than they are a bacterial cell. And they are really different from bacteria, other than in size. Okay. So your membrane area has a lot to do with what a cell can do. And prokaryotes don't have membrane-bound organelles, but they can use their plasma membrane infolded to get a lot of the functions that we associate with our organelles, like energy production, in a prokaryotic cell. So we typically think of prokaryotic cells as being inefficient in terms of generating energy because they don't have mitochondria, they don't have chloroplasts, but there are prokaryotic cells that act just like mitochondria or chloroplasts by just folding their outer membrane inside the cell over and over and over again. And I'll show you a picture of that. So membrane area is really important because you have to deliver anything that's coming into the volume of the cell through that membrane. And so you want to keep the area of membrane per volume of cell that you've got to serve at a certain ratio. If you take a cube of a given size and you divide that into lots of small cells, a little bit of a few cells, or one giant cell, they all have the same volume. But as you go toward one giant cell, you have a lot less membrane per area that it has to serve, or per volume that it has to serve. Okay, so let's say if you have in this volume, you have 1,000 different cells, then the total surface area supplying those 1,000 cells of 8,000 cubic microns is 24,000 cubic micrometers, or a surface to volume ratio of 3. If it's just 8 cells, you have the same volume, but much less surface area supplying that volume, and a much lower surface area to volume ratio. And then one giant cell, you have, again, the same volume, but a much less, 10 times less surface area of membrane supplying that volume, and a very low surface area to volume ratio. So since everything is coming in and going out of that cell through the membrane, you need a lot of membrane supplying whatever volume you've got to serve. Here's an example of a prokaryotic cell that has taken the adaptation of just taking its outer membrane and folding it repeatedly on the inside to get all kinds of membrane surface area. And that membrane surface area can perform photosynthesis very similar to what a chloroplast would do, even though this is a bacterium and not a eukaryote. So here's a H5P question for you. Okay. So here we're going to just walk through some of the organelles in a cell, and I'm going to give you generalized pictures of these things. I want you to know, like from one of your first podcasts, that there's tons of diversity out there in cell shape, and you shouldn't take these diagrams too seriously in that this is what represents a true prokaryotic cell. Prokaryotic cells are so diverse that we don't even know most of the species that are out there. And so we certainly haven't plumbed the depths of shape and size that's out there. But let's just say you had a general prokaryotic cell that was about 10 microns long. It had a cell wall on the outside, a cell membrane inside that cell wall, and then a circular piece of DNA that was all wrapped up. Ribosomes that are floating in the cytoplasm that can do protein synthesis. No membrane-bound organelles. One more thing about this. In addition to the giant circular chromosome that's all folded up, there can be an extra chromosomal piece of DNA called a plasmid that can carry genes and do functions for the bacterium. A lot of antibiotic resistance genes are carried on plasmids. And in the lab, molecular biologists use plasmids to transfer among bacteria to give them different properties all the time. So in addition to the giant chromosome that carries the DNA to make the bacterium when they reproduce, there can be these extra circular bits of DNA called plasmids commonly in bacterium, in prokaryotes. If we look at a generalized, so this could be an archaean or a bacterium, okay? If we look at a generalized animal cell, again, lots of diversity among animal cells. But if we just say your general animal cell, what does that look like? And what would it have in there? There are many organelles contained in that cell. An animal cell would have an outer plasma membrane, also called cell membrane. They're the same thing. And would not have a cell wall. And then many membrane-bound organelles inside. There would be a nucleus that is bound by a nuclear envelope, a double membrane system that is contiguous with the endoplasmic reticulum system. So you can see here in this diagram that the nuclear envelope, the membrane, double membrane system that covers the nucleus, is continuous, okay, becomes part of the endoplasmic reticulum. So those systems are connected together. Okay, a lot of people sometimes miss that point. Then there's another membrane system called the Golgi apparatus. It has a cis face and a trans face, and that proteins move through the Golgi apparatus through vesicles and then will fuse with the plasma membrane in the endomembrane system. We'll talk about mitochondria separately. Within the nucleus is the DNA, and then you can have a region called a nucleolus, which is not membrane bound and is dominated by RNA within the nucleus, and there can be more than one nucleolus within a nucleus. We'll talk, like I said, we'll talk about mitochondria. Lysosomes are low pH organelles where debris from outside the cell and or old organelles will be transported to the lysosomes, broken down by digestive enzymes, and recycled into their macromolecule components, amino acids, nucleic acids, and lipids and protein, nucleic acids, amino acids, lipids, and carbohydrates. Within the cell, if a protein is at the end of its life, it does not go to the lysosome. It goes to a not quite an organelle, but a very large structure in the cell called a proteasome, and that's a different system. Peroxisome, which looks like the same sort of thing in this diagram, and they can be difficult to distinguish from lysosomes, except that peroxisomes tend to have very high concentration of an enzyme called catalase. Catalase is a detoxifying enzyme that deals with antioxidants, and so it detoxifies peroxide radicals, and sometimes the concentration of catalase inside a peroxisome is so large that on an electron micrograph you'll see it as a crystal, and it looks like a perfect square within a peroxisome. So that's a dead giveaway for a peroxisome. Peroxisomes, in addition to mitochondria, can break down fatty acids in beta oxidation. We'll talk about that later. And then there'll be hundreds of thousands of ribosomes and cytoskeletal elements that we'll talk about later also. And then a centrosome is a microtubule structure that is seen and plays an important role during mitosis, and we'll talk about that later. If we characterize a generalized plant cell, a couple things that would be added in addition to the animal cell. Of course, there's going to be a cell wall, a cellulose cell wall on the outside of the cell membrane. So the cell membrane is on the inside, cell wall is on the outside. The cell membrane is on the inside and still fluid mosaic and all that stuff. You just have a rigid cell wall on the outside. The plant cells tend to have a huge water-filled vacuole, central vacuole, where they maintain part of the plant skeleton by, if that is full of water, then the plant is standing upright, turgid, like celery, stalk of celery that you could snap, versus if there was not much water in the central vacuole, the celery would be flaccid and bendy and be hard to snap. Of course, chloroplasts are a big feature of plant cells. Animal cells don't have chloroplasts. Chloroplasts have 3 membrane systems, and we'll talk more about those when we talk about photosynthesis. And then plant cells have connections among them that are these long, thin connections through the cell wall, where one cell can communicate with another cell, and that's called plasmodesmata. If the cells are giant, like we talked about neurons being really, really big as an example of an extreme cell in an earlier podcast, a eukaryote will use a transport system to get around the problem of diffusion. So we talked about on that long, boring table about why it is you want your membrane to surface area to volume ratio to be high, so that you can get membrane supplying just a little bit of your cytoplasm. If you're a giant cell, that's a real problem. And they get around that problem by using specialized transport systems, such as here is an example of a freeze etch micrograph where you see a microtubule track here, and then vesicles on that track carried by motor proteins, and these vesicles can be even whole organelles. So later on we'll talk about this system where you have a motorized protein that uses ATP and GTP to move things down a cytoskeletal network like an axon and get over the problem of diffusion, limiting cell function in a very, very large cell. They can also use something called cytoplasmic streaming, where there is a motor molecule that is physically moving the cytoplasm around, not just relying on diffusion to move molecules around in the cytoplasm. And there are also these super enzyme complexes, whereas if one of those biochem or principles of biochemical pathways that you memorize, where you said in step 1 this thing binds to that thing, and in step 2 it binds to this thing. In superenzyme complexes, those enzymes are physically attached to each other, so that you have the product of one reaction being immediately available as a substrate for the next reaction. Because you don't have to rely on diffusion to get to step 2, because they're physically attached, that can get around some of the problems of big cells. One characteristic of eukaryotic cells is that they have linear chromosomes. There are some exceptions of this in some archaeans, but in general, archaeans have circular chromosomes and eukaryotic cells have linear chromosomes. You would only see chromosomes that look like this during cells that are getting ready to divide, getting ready for mitosis, and see individual multiple chromosomes per cell. That is also a eukaryotic feature. In prokaryotic cells, you typically have one circular chromosome per cell. Some pictures, micrographs that go along with those pretty diagrams that we just saw. If we look at the nucleus in a transmission electron microscope picture, this is where you take a thin slice and you stain it. The nuclear envelope is out here, this double membrane. Chromatin, this is the DNA plus histone protein that the DNA is wrapped in. Comes with a speckled sort of feature. The nucleolus, the RNA-rich area with no membrane around it, stains dark. And then nuclear pores are where things enter or exit the nucleus. In a freeze etch, freeze etch picture, where or freeze fracture, that's what I'm trying to say. Freeze fracture picture, where you take a nucleus and you break the cell, you freeze it, and then you break it. It'll break on those double membranes, leaving one half of the membrane system on one half of the break and another half on another half of the break. You get to see these nuclear pores pretty dramatically in the nuclear envelope, where you're just looking at one half of it. So the chromatin during mitosis, you would see that as chromosomes. This is not during mitosis, so it's diffuse. The nuclear envelope is continuous with the ER. Remember, they're connected. I showed you that on that diagram a couple slides back. The nuclear pore is where processed mRNA is going to leave the cell to be translated, or leave the nucleus, sorry, to be translated in the cytoplasm or on the endoplasmic reticulum. And the nucleolus is dominated by ribosomal RNA. There's no membrane around it. Okay, mitochondria. Spend a fair bit of time talking about mitochondria, so-called powerhouses of the cell. Two membranes, an outer membrane and an inner membrane that's all folded up. The inner membrane folds are called cristae. The innermost compartment, so inside the outer membrane and inside the inner membrane, is called the matrix. These two membrane systems have a different protein component. The protein concentration on the inner membrane system is really high, up to 90% protein by weight. When I ask you to draw a mitochondrion, this is what I want you to do. Okay, I want you to, let's see if I can get this pen. Here we go. I want you to just draw one line per one membrane. So here's one, and then here's your inner matrix protein. So one membrane, two membrane, and then here is the matrix, the innermost component. I don't, so always for a test in my class, one line equals one membrane. Don't draw two lines because you're trying to show me the two halves of the leaflet of a membrane. Don't do that. That's just confusing to everybody. One line is one membrane. Also, I don't want you to draw this. Some people try to draw a 3D version where you can cut away part of it and see the cristae. I don't want you to draw that because, frankly, you're not that good at it. Just draw a simple diagram like this, and I will know that you mean that's the outer membrane and that's the inner membrane. The space between the outer and the inner is the intermembrane space, and the space all the way inside is the matrix. Okay, mitochondria have ribosomes inside of them. Mitochondria have transport systems between the outer and the inner membrane, and between the outer membrane and the cytoplasm. And mitochondria are a large anastomosing membrane network. They're not little kidney beans like you see in all these diagrams in all the books all the time. So when you see a picture like this, which is a transmission electron micrograph, they fixed that cell using chemicals, and then they sliced very thin through that cell. And so you're seeing a very thin slice through part of a mitochondrion. But that mitochondrion is connected to a whole network of mitochondria. So if you imagine your hand, and if you took a very thin slice through your fingers of that hand, trying to image that hand, you'd see the profile of all your fingers, and they would look disconnected, and you would not know necessarily that it was connected through the rest of your hand. Kind of a gross analogy, but hopefully that gives you the picture. So in reality, mitochondria are changing shape constantly. So I'm going to try to show you this. I think here is, yeah, it's like a lava lamp. Okay, so here's the mitochondrial network. It's fusing and then breaking apart and then fusing again and then breaking apart. And you have individual mitochondria that might break off and then fuse and form one big mitochondria and so on. It's constantly changing all the time. Now, if I just froze this at one point in time and sliced through here, I get a profile of this and a profile of that and a profile of that, and they would look like individual mitochondria. But the reality is this is what's going on inside your cell. One little important consequence of mitochondrial function is that in males that make sperm, the mitochondria are concentrated near the head of the sperm, and only the head of the sperm will penetrate the egg. And the mitochondria have their own genome. They have a circular genome, just like prokaryotes, and they probably were prokaryotes. Talk about more about that in a second. But that genome from the man isn't transmitted into the egg. And so your mitochondria, if you're male or if you're female, came from your mother. The mitochondria in the egg of the mother are what's passed on to the next generation, not the mitochondria of the father. Now, there are some very interesting exceptions that have been identified in the last couple of years, where there have been some mitochondrial diseases transmitted through the father, and that could only happen if indeed the father contributed his mitochondrial DNA to his children. These are thought to be super rare, but it can happen. In general, the idea is that you get all of your mitochondria DNA from your mother, not your father. Chloroplasts have 3 membrane structures, 2 outer membranes, 1 inner membrane that's all folded up. The inner membrane system, when it's all folded up, that third inner membrane is called the thylakoid membrane. A stack of thylakoid membranes on the innermost space of the mitochondria is called a single stack is called a granum, and plural stacks are called grana. And these sort of look like pancakes. Who makes the best pancakes? Grandma. So you can remember grana, grandma. Okay. 3 membranes for a chloroplast. Again, if I ask you to draw a chloroplast on a test, you're going to draw 1, 2, okay, and then you'll have this stack. Sorry, I'm drawing this on a mouse, so it's worse than it would be normally. Okay, 1, 2, 3 membranes. These shouldn't be touching each other, but that's the best I can do right here. And 2 outer membranes, 1 inner membrane, all folded up. This third inner membrane is called the thylakoid membrane. A single stack is called a granum. Plural stacks are called grana. The space inside that third membrane is called the lumen. And the space outside that third membrane, but inside these 2 membranes, is called the stroma. Okay? So plants have mitochondria in addition to chloroplasts. Don't forget that. But animals don't have chloroplasts. There is one very cool exception. That is this Alesia chlorotica. It is a sea slug or a sea hare are the common names. A nudibranch is the scientific name. It's like a snail without a shell. You can see these guys in the Florida Keys in the mangrove swamps. And what they're able to do is they're able to eat algae and take apart the cells of those algae, but not digest the chloroplast. Instead, they migrate those chloroplasts to their backs. That's why they're green. And they can actually photosynthesize using chloroplasts that they stole from the food that they're eating. And the animal is actually able to harbor these chloroplasts and derive nutrients from the photosynthate that the chloroplasts make. That is pretty remarkable. There was a couple papers back about 10 years ago saying that the chloroplast DNA had been transferred into the genome of the animal, but then nobody could confirm that, so that's still out there to be decided. Okay, here's an electron micrograph of a chloroplast. Two outer membranes, you can't see them as separate membranes unless you really zoom in, but there are two out there. This is the inner thylakoid membrane. Here's a stack, granum, and then several stacks called grana. Okay, chloroplasts also have ribosomes in them. The light reactions of photosynthesis happen on the grana. The dark reactions, the Calvin cycle of photosynthesis, happen in the stroma, which is the space outside the third membrane but inside those two outer membranes. Okay, here's another H5P question for you. And here is a bonus question. So if you are early to the podcast this week and you're one of the first three people who can think about this and get me the correct answer, just send it to me by an email. I will add 1 point to your first exam. As soon as I get 3 people who have the right answer, I will tell the class. If you get it wrong, I'll just tell you it's wrong and you can try again. Okay? Okay. So, I mentioned before that mitochondria have their own DNA and it's circular DNA. Chloroplasts also have their own DNA and it's circular DNA. We know from that long, boring table slide that circular DNA is a characteristic of both bacteria and archaeans, characteristics of prokaryotes. So there is strong evidence that chloroplasts and mitochondria were once free-living prokaryotes and or archaeans, and that they were engulfed by a eukaryotic cell. Instead of digesting those components, the cell kept these organelles and then were able to take advantage of them. Mitochondria in terms of making ATP, chloroplasts in terms of making ATP and making food. So in addition to the circular DNA, chloroplasts and mitochondria are about the same size as prokaryotes, and they have membrane systems as if they got engulfed. So if they engulf this, they would be covered with an extra membrane. Chloroplast has 2 membranes, mitochondria have 2 membranes, and chloroplasts have 3 membranes. So not 1, but 2 engulfing events. Okay? Maybe other organelles were also once free-living. Not as strong evidence for those, but very strong evidence for chloroplasts and mitochondria. That's why it's called an endosymbiotic theory, not the hypothesis, but a theory, in that this is an idea that has withstood many, many experiments and stands up to lots of data. And the best explanation for all those data is that they were once free-living prokaryotes and now are part of our cells living symbiotically. Okay. Moving on in our walk through the organelles, we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The endoplasmic reticulum is connected to the nuclear envelope. The difference between rough and smooth is rough is studied with ribosomes, smooth is not. Smooth and rough have different functions. Smooth ER is involved in making membranes. Rough ER is used in making proteins that are going to be part of an organelle, membrane bound, or exported out of the cell. So any protein that is going to be part of an organelle, or is going to be membrane bound protein, or is going to be exported out of the cell, like all of the protein hormones that come from, say, your pancreas cells or your fat cells, and exported into the blood. All of those were made on the rough ER. The only proteins that are made on the cytoplasmic ribosomes are proteins that stay in the cytoplasm. Let me say that again. The only proteins that are made on cytoplasmic ribosomes are proteins that stay in the cytoplasm. So most of the protein in a cell, that's not the case. Most of the protein in a cell associated with a membrane or an organelle or is exported outside the cell. In that case, it's all made on the rough ER. And this just shows you how it's continuous with the nuclear envelope. On a transmission electron microscope, you can see the individual ribosomes on the ER. There are hundreds of thousands, if not millions, of ribosomes in a cell. Depends on the size of the cell. But lots of ribosomes on the side of the cell, inside the cell. And again, these micrographs are only a slice in time. So the tissue is fixed at a certain time, and then you take a picture of it after you slice it. These ribosomes in real time move on and off the surface of this membrane as part of the translation system. And we talk about that in cell phys. We don't talk about it so much in this class. But it's only rough because those ribosomes have to associate with the membrane when they're actually translating the protein. But as the mRNA first is read by the ribosome, it's a ribosome that is sitting in the cytoplasm, and there's a tag on the mRNA that says to the ribosome, go to the ER. And there's a protein system that takes it to the ER, and that's when rough ER becomes rough. Rough ER delivers its proteins to the Golgi apparatus. The Golgi apparatus is named for the person who discovered the system in eukaryotic cells. It has a cis face facing the nucleus and a trans face facing away from the nucleus. Their proteins get modified and then moved into different flattened sacs called cisternae as they move through the Golgi apparatus, and they're biochemically modified as they move through the Golgi apparatus. If a protein is glycosylated, it has that carbohydrate has been added to the protein in the Golgi apparatus. There are other types of protein modifications like meristylization, etc., that will happen in the Golgi apparatus. Proteins will move in a vesicle from one stack and to another stack as the vesicle blobs out of one stack and fuses with the stack up above it. And there are specific mechanisms on the outside of these vesicles, other proteins, to recognize where it's supposed to go and if it's supposed to dock there or not. Okay. I've mentioned the endomembrane system several times. We're going to be getting into it more. The default endomembrane transport system is that a mRNA comes out of the nucleus. It would be started to be translated on a cytoplasmic ribosome. A signal would then take it on, a signal that is part of the mRNA would then take it to the rough endoplasmic reticulum. That protein would be synthesized. It would be packaged into a vesicle. That vesicle would fuse with the cis face of the Golgi apparatus. It would be modified or not. and then it would be repackaged into a vesicle, fused with a medial face, and then eventually the trans face of the Golgi apparatus, moving toward the plasma membrane, and eventually packaged into a secretory vesicle, which would then fuse with the plasma membrane and be exported outside the cell. If a signal that is part of the mRNA does not tell the protein to do something else, this is what will happen by default. So if there's a signal on the protein saying be translated on the rough endoplasmic reticulum and then no other signal telling it where to go, it will end up being exported out of the cell. Other signals that it could get along that is also part of that original mRNA could be, say, go to the inner mitochondrial membrane, or go to the lysosome, or go to the plasma membrane, or go to the nuclear envelope. Those are all possible offshoots from this pathway, but if those extra signals are not on there, the default for any protein that is translated on the rough ER is going to be squirted out into the blood. And again, cytoplasmic proteins are made on cytoplasmic ribosomes. So if there is no signal saying go to the rough ER, you're translated on a cytoplasmic ribosome and you stay in the cytoplasm. But any other compartment in the cell, any organelle, or even outside the cell, you're going to get a signal telling you where to go. And that signal will be on the mRNA. Okay. The lysosomes that we mentioned before are a low pH environment inside the cell. They degrade stuff that comes from outside the cell and or organelles that are being recycled within the cell. So if there's a protein being recycled within the cell, it does not go to the lysosome, but if it's a whole organelle, it will. And stuff from outside the cell will be degraded inside the lysosome. So how you import cholesterol into your cells is there's a big ApoB protein that has a big ball of lipid, including cholesterol in it, that's recognized on the outside of the cell, taken into the cell, and then fused with the lysosome, and the digestive enzymes in the lysosome breaks apart that protein ball into amino acids, cholesterol, and lipids. And then those are used by the cell after they go through the lysosome. In the transport of things going through the pathway, the endomembrane system pathway, there can be tags on proteins that get put on the cells along the way that will help them reach their normal functional compartment. So proteins that end up in the lysosome have a problem in that the cell has a problem in that these are proteins that take apart other proteins. They're proteases. And so if the whole cell has protein all over it, and you're making a protein that takes apart other proteins, then it could potentially take apart the system as it travels through the system. But instead, there is a carbohydrate tag called mannose 6-phosphate, mannose 6P. Mannose is a carbohydrate that has a phosphate on its number 6 carbon. This tag acts like the little piece of plastic that you get between the two batteries when you get like a remote. So that in the store, if you press the remote, nothing happens because the batteries aren't in contact with each other. And then you pull out that little slip of plastic, and now they are in contact with each other. Mannose 6-phosphate prevents the enzymes from being active until they reach this late endosome lysosome compartment where the pH is low. Once they hit that low pH environment, then the phosphate comes off, and these proteins can do their job digesting enzymes in the lysosome. But while they're traveling, that phosphate is still on there, and they don't digest anything. Okay? There is something called eye cell disease, where there is no tag, and instead of going to the lysosome where it's supposed to go, these hydrolases are secreted via exocytosis, and they can cause damage outside the cell because their job is to break down protein. Okay, this is just an example of a cell transmission electron micrograph that has a picture of some lysosomes that are unusually large. They have a lot of components in there that's characteristic of a lysosomal storage disorder. This is not typically how cell lysosomes look like. It's just an example to show you what a lysosome would look like if it was huge. Okay, peroxisomes, as I mentioned before, can be recognized because sometimes they have a catalase crystal inside them. And this is just a very high concentration of this enzyme catalase so that it crystallizes out in the fixation process. And you can see that and identify it as a peroxisome. Otherwise, it's just another circular thing with gray stuff inside of it. But this crystalline core is for catalase. Catalase, again, detoxifies peroxide radicals and also can do beta oxidation fatty acids in addition to mitochondria. And then the giant vacuole that you see in cells is just a large fluid-filled membrane component that can get bigger or smaller depending on the water availability to the plant. There's the cytoskeletal network that keeps all the organelles in place and also helps aid movement of molecules through the system and gives a cell shape. It is made up of microtubules, which are the largest diameter fibers that are made out of tubulin, microfilaments, which are the smallest diameter fibers made out of actin, and then intermediate filaments, which are made up of a whole range of proteins that are in between the diameter of a microfilament and a microtubule. These are all just labeled with 3 different fluorescent stains and an antibody so you can see them all in the same cell. This big area here is where the nucleus would be. And you can see how these filaments permeate the shape of the cell and then really fill up the cell. I often say that you should think of the cell and its organelles like a soggy Nerf ball, like it's a solid thing, and that all those spongy connections in between your Nerf ball are like the cytoskeleton made up of microtubules, microfilaments, and intermediate filaments. And that it's really packed full of space. There's not a whole lot of liquid to swim around in there. It's not like a bag of vegetable soup. It's much more like a soggy Nerf ball. Also important are these plasmodesmata. These are connections between plant cells that go through the cell wall that allows one plant cell to connect with another plant cell so that the cytoplasm of the 2 plant cells are connected. Okay, another H5P question for you. And that's it.